we can then I will share my screen so that we can talk about the plant hormones or the plant rooting hormones that we've been using. So I mentioned before that plant hormones are not really necessary. So rooting hormone is not really necessary because it is a modern day development. Even though propagation, or most of the techniques of propagation that we're doing right now, they are over 4,000 years of age. Back in the day, they were still able to root a lot of plants and get a lot of success without the using of the rooting hormones that we have right now, or the rooting powders. So is it help? Yes. Is it speed up the rooting process? Yes. And so that's why we continue to use them. Different nurseries are going to have their own preference for the different rooting hormone. So some of them are going to like the liquid, some of them are going to like the powder, and some of them may like some kind of gel. And as they experiment with different plants and experiment the different percentages or strength on the rooting hormone, they are able to propagate many different plants right now. And the ones that they are not successful, they are working with them until eventually they figure out how. Probably one of the best examples is going to be the olive tree. So for a long time they couldn't propagate it, and then they came up with a variety that was seed fruitless, that supposedly is going to bear less fruit because they're kind of messy. So just through experiment, what they need to do is that they will take the cuttings, they will then let them sit overnight in a liquid rooting hormone that is high percentage of the auxin. And by letting them sit in the rooting hormone for many hours, then they strike them, and they were very successful. And that's how now all of the all of the olives are propagated through that method, especially the fruitless olive. So does the percentage of the different hormones influence the rooting? The answer is yes. And it's going to be a lot more important when we carry out the micropropagation or the plant tissue culture or plant cloning in a test tube, because then micromillimeters of a hormone can have a good or bad effect on your results. But like us, the plants have many hormones, and when they work together, it makes possible for the plant to carry out its life. And when they are out of balance, then it can throw the plant out of whack. Same thing will happen with people. So rooting hormones, we're going to find, again, just a little bit of, again, a review. So we have just a general drawing of the plant. And so we have the rooting tip or the shoot tip that is going to be responsible for many aspects of the plant, including the dominance. So the tip of the plant always want to have dominance because it wants to be the highest most point, and it's going to achieve that by sending out a hormone that will keep the lateral buds dormant. If something happens to that tip, then the buds begin to wake up and they fight for the dominance. And then we have some of the leaves and the vascular system that we talked about before, and then the roots and the root hairs and all the other parts. And so very important when we are dealing with a plant hormone is that a plant hormone is going to be produced in an area, let's say it's going to be created in the apical meristem of the plant or the tip, and then it needs to be located or translocated via the vascular system to a different area of the plant where it's going to carry out the change or the effect that you want it to, or the normal work that that hormone needs to happen or occur or bring to that specific plant. So when we're talking about the products that are going to be naturally occurring on plants, we're going to refer to them as plant hormones. And so we're going to have just organic compounds that are going to be produced in the plant and then taken somewhere else where they create the change. But when we are going to be referring them to a product that is then synthesized in a lab, then those are going to be called plant growth regulators, because as we have now studied the chemical makeup of each of those hormones, now they are going to be produced in a lab, and now those are going to be the product that you are going to be using when you use the rumen powder. So are you using a plant hormone? The answer is no, unless it came from the plant, but most often it's going to be now a growth regulator. So that's going to be, if it comes from the lab, it's a growth regulator. If it comes from the plant itself, or if it's made by the plant, then it's a plant hormone. And we use them in several aspects other than propagation. We can use certain hormones to abort flowers and prevent olives and other plants from having a lot of fruit that could be very messy. We also use growth regulators to stunt poinsettias, for example, so that they're not too leggy and they don't break as you pick them up in the nursery and take them home, and also chrysanthemums and a bunch of other things. So there is a slight use for them in the horticulture industry. And so when we look at the major type of hormones, there's going to be auxin, cytokinins, and gibberellins. Should you know these hormones? The answer is yes. You should know the names, but don't worry about the chemical makeup. Don't worry about too extensive about what it can do for the plant, because they're all going to have a very important job. They're all going to carry out or allow the plant to carry out its biological function, and they're all going to keep each other in check. So just be aware of them, know the name, so that when you see something or when you see them in your reading, that you know what they are talking about. And there's going to be ethylene and abscisic acid, which are going to be the other two. Ethylene and abscisic acid are going to be also known as the destruction or the destroying hormones. So these are going to have a negative consequence on the plants. So these are the five. Again, be familiar with them, know the names, but that's about it. Don't worry too extensively as to the chemical makeup, even though I'm going to show it to you right now. But this is not a chemistry class, and unless you're going to dive deep into tissue culture, by that time you probably will need to take an extra class on advanced plant propagation, where you would have to study that a lot more. But for our purpose, just know the five different hormones and what they're going to do. So the first three are going to be the growth promoters, and the second or the last two is going to be the growth inhibitors. I mentioned the destruction would be caused by the letter two. And so when we look at auxin, auxin is going to have a very, very important role. This would be the chemical, the hormone that will keep the dominance to that apical meristem or the apical bud. And it was a very lengthy process to where people just found out about the hormones. So they knew that there was something within the plant that would cause it or need it to occur or bring about changes. So auxin is also associated with phototropism, which is the ability of the plant to see the light or to follow the light. And that's why when you take a plant and you put it inside the house, it will always grow towards the light, because somehow the plant is able to see the light. And they couldn't understand how or why, because obviously plants do not have eyes. So they figured that something within the growing tip of the plant had to be responsible for allowing the plant to see the light and making the plant grow uh, towards the light. And so the control plant on the far left obviously has nothing happening to it, so it started leaning towards the light. And so the second one, when they removed the tip, there was no movement towards the light. So that's how they found that it was at the tip of the plant that whatever is happening, it's going to come out of there. And by removing the tip, then you prevent that from happening and nothing happens. put a cap that did not allow light to reach the tip of the plant, and the plant once again did not move. So something at the tip that is going to be triggered by the light is causing that. And then when they put a clear cap on the tip that allowed for the light to penetrate, then once again it moved towards the direction of the light. Now they also had to prove that it wasn't something that occurred maybe at the bottom or maybe at the roots or somewhere below. So they put a cap or they covered the base of the stem, and obviously the plant started leaning. And then they also had to make sure or figure out how uh, if the product was water soluble and it was able to penetrate through. So they cut the plant or the tip, and then they put the gelatin, and then they put it back through, and the plant started to grow because the hormone was able to go through the gelatin agar block, and it was able to bring about the change. And when they put a mica that was obviously not water soluble, they prevented that from happening, the plant didn't move. So these were all the different experiments that had to take place. And the final one was when they took the tip and they put it on a agar or a piece of gelatin. The hormone went from the tip into the agar, and then later on, when they put that agar on a cut piece of the plant, it brought about the same change as if the plant had the top or the apical meristem. And so here you have a different method. So it was a hormone or a something that was made at the tip that was water soluble and able to go through the gel, and it would be causing the stem to reach and bend towards the light. And that's how they saw kind of figure out how some of those hormones work. And so when we look at Auxin, these are going to be the types that we're going to find. So when you read the label on the rooting powder that we have here, it's going to have this word IAA, indoleacetic acid, and that is just a form of that synthetic auxin that we have in horticulture. There's also it's broken in light, which means that once you have taken it out of the container, you don't use it again. So that's why you only use it for the day. And I usually chuck anything that remains because it's also contaminated. So make sure if you're going to have some that you don't allow it to see the light because it starts to break down. It's some of the synthetics that we have are going to be IBA, indoleabutyric acid, or NAA. So these are just going to be the different forms. So when you read the label, different companies will have a different mixture of some of these products, but they all have the goal of creating the roots. So this would be the hormone that is going to be responsible for generating the new roots on your cuttings. Uh, and also 2,4-D, which is a pesticide. This was Agent Orange that was used in the Vietnam War. So the way that they were able to deforest an area was by adding a lot of hormones that would literally kill or make the plants grow to death. They would grow so fast that they couldn't handle it and they'll die. And so that's how the work of this pesticide, because it's nothing more than a synthesized rooting hormone that brings things out of whack. So the site of the plant that makes the auxin, so mainly the shoot tips or the root tips. And so that's going to be very important for us. So shoot tips and root tips, also young and expanding leaves. And that's going to be also important for us when we do the leaf cuttings. So we don't necessarily need to add a rooting hormone, but we will, because if we don't, it will have some auxin, natural auxin out of the leaf. Uh, and also the young seeds because it's going to have a valuable role on the development and also the germination of the seeds. So, and it plays just a general role of making sure that the plants works well. So so where from the Auxin promotes or inhibits the leaves, the fruit, and the flower. So this is where if added in very large dosage, it will cause the flowers to abort and or the leaves to drop. And so the removal of some of these hormones by blocking it from a different one and just not having it around is what's going to and the oncoming or the onset of that cold temperature. So the hormone applied to the blooms of an apple are going to thin out. So in many cases, growers do not want to go through or spend money on employees to go thin out the blossoms on an apple. And so they will spray the trees so that the tree will naturally abort some of those flowers. And so that would be a way that they would save on their workers. And then applied to young fruits also exhibit the dropping of the small apples. So it allows them to remain longer in case the tree wanted to naturally thin them out as well. Uh, the concentration determine stimulatory or inhibitor system. So in very high amounts, the auxin can have an inhibitor or it can inhibit or cause a problem. So one of the things that I asked for you to do is that you are going to dip the cutting and you're going to tap out or tap off the excess hormone. Because if you have a blob of hormone on the root, on the base of the cutting, it's going to inhibit the production of the roots. So that's why that was the reason why I asked for you to do that. You just need to tap it a little or just dip it a little, but don't swirl it around. Don't allow for a large amount to remain because then it's going to have a negative consequence or a negative impact on the plant. In low concentrations, it promotes the roots, the adventitious roots that we want, and in high concentration, it's going to prevent them from occurring. 2,4-D is a herbicide. And so Auxin also has the ability, as mentioned before, of phototropism. So phototropism is the movement of the seeds or the plants towards the light. And it will happen with plants that are going to grow in dark areas, or if you keep them indoors, it is also recommended that you turn the plants every so often so that they're not going to be growing towards one side and kind of be one-sided, because they will follow the light because that's what they need to survive. So that's going to be the Auxin. Geotropism is going to be the growth of the plant in respect to gravity. So this is what's going to allow the tip of the plant or the shoots to grow up and the tips of the roots to grow down. So no matter how you plant the seeds, you can plant it upside down, right side up, on its side. The shoots are always want to grow up and the roots always want to grow down. So it's a combination of gravity and some of the hormones that allow the plant to figure out which way is down and which way is up. So at the tips of the roots, there are some starch grains. And so in the roots, when the starch are going to be going towards the bottom because of gravity, that's the direction that the seed, the root is going to grow towards. From the roots, they're going to be at the bottom. And when they're going to decide how to grow up, the same starch are going to be at the bottom respecting gravity. So the shoot is going to grow opposite to wherever the starch grains are. And that's always going to tell them how to grow up. So starch grains, hormones, it's what's going to allow the plants to grow, because then the hormone is going to be directed to the area where the stem is going to begin to grow up and the roots are going to grow down, and or bend the stem upward and the roots downward. So geotropism, plants respect gravity, plants know which way is up and which way is down. So here's just a drawing of the concentration or how much is produced by the plants and which location. So we know the apical tip or the apical meristem, the tip of the plant is going to have one of the highest. The same thing with the roots, because that's what's going to allow them to grow. And then as you see, it starts to diminish until somewhere in the middle, it's just going to have a small amount. But by far, the biggest concentration or the highest amount is going to be at the tip and of the shoots or the apical meristem, or the lateral buds, if they also have to be the end buds, or the roots, and also the lateral roots will be the same. So additional things with accent, so it's going to allow the dropping or the loss of the leaves naturally, so it's going to create a barrier. It's going to initiate the flowers, so during the right season, the right amount of auxin is going to begin the development of the flower buds. It could determine the sex on the flowers, and or they can be changed. That will be the auxin as well. It also is going to have a good influence on the development of the fruit. Uh, and uh, uh, and so here is what's going to happen when auxin is going to be present in high amounts inside the leaf is going to be happy, the leaf is going to carry out its function. But as soon as the temperature begins to change, starting to get cold, then the auxin is going to be diminished, and then the abscisic acid is going to become a lot more. And what that's going to happen is that then the hormone or the changes in hormone is going to create a natural break in the leaf. And that's going to then drop the leaf and allow it to fall to the ground and be recycled. And the next year, the tree is going to make a brand new root. So it's no harm to the plant. It naturally creates a breakup point and it drops the leaves naturally. So it is the combination of the auxin and the abscisic acid and the other hormones that will play a role in that. When we do leaf cuttings later on, here's the African violet. So the auxin that it will be produced by the leaf, so we want to make sure that we leave the blade, is going to be translocated to the cut or the base of the petiole, and that is where we're going to have the cells that are going to revert to being an embryo or revert to being a root, and we're going to have a brand new plant. We're going to speed up the process by adding the hormone at the base, but if we don't add it, we can still get away with propagating through leaf cuttings. So that's the leaf that also produces a small amount. And here's the cutting that we are making. So the auxin produced in the buds or the apical buds or in the lateral buds will be translocated into the wounded area, and that is what's going to initiate the new roots. And that is why, that is the reason why grandma was able to root many things in water, because it will be the auxin that will naturally occur in the plants that will cause that. We have now the modern day hormones, the modern day growth regulator that allows us to do this to plants that are a little bit more difficult, that grandma would probably not be successful or not be as successful. And it is why nurseries can grow 1,000,000 plants in a year without any problem, and they could be very successful with this. So that's just the added bonus. So the auxin is important for the seeds. So what happens is that when pollination and fertilization takes place and the development of the embryo and the seed begins, the seed or the embryo is going to put auxin into the receptacle, and that's what's going to allow the strawberry to grow, and or what's going to allow the development of the fruit, apples, peaches, and apricots. So it is the embryo that has to be alive for this to happen. When they remove the seeds from the strawberry, there was no site for the hormone, so then the receptacle did not grow. But when they removed the seeds and sprayed it with a artificial auxin, then the strawberry grew like normal, proving that the seeds and the embryo does play a role in the development of the fruit. And if for some reason a fruit or the embryo gets aborted, then we know that fruit could be lopsided and they don't develop properly because they stop getting the right hormone or the right hormone for the proper development of the fruit. So that's when you get distorted fruit or ugly fruit, as they're referred to, not the perfect ones. Agent Orange, the hormone, is going to stimulate the very rapid multiplication and division and elongation of the cells. And so when sprayed on the plant, it's going to grow and multiply so quickly that it's just going to be very spindly, and the plants are forced to grow, and they grow themselves to death. And that's how it works. It's still used in some of the herbicides, some of the, not Roundup, that's a different product, but some of the other herbicides that are not glyphosate based will still use some kind of growth regulator to help kill some of the weeds. But we know that Agent Orange it's been now linked to a bunch of problems with veterans, and we don't like to use it here. But it is a normal, or at least at one point it was a normal plant hormone. So then the next one is Gibberellin. So once again, don't worry about the chemical formula. Gibberellin is going to be was discovered or is known as fool's disease or foolish disease. It is caused by a fungi, and it is first discovered with rice. And so what happens with this hormone is that it's going to be responsible for the elongation of the plants. And so plants that were infected with this Gibberella fungi, they were being injected with extra hormone by the fungi that kept expanding and elongated the cells of the plant, where they become very spindly and very leggy. And then the plants would become so leggy and spindly and tall that they would fall to the ground. So they were very full because they could not stop growing. And that's why they call it foolish diseases. So later on, they analyzed the fungi, and they found that it was the fungi that was creating some kind of plant hormone that was triggering this kind of growth on the rice plant. And so that's where Gibberellin was discovered. That's how it was named, after the fungi. And so we have some of the Gibberellins used in the plants. So different types that are going to be made by the plants. So the large complicated molecule, which means that they have not been able to synthesize it in the lab. So they still are producing it naturally from the fungi. So they harvest the fungi, they culture the fungi in labs, and then they extract the Gibberellins from it. And that's how we are able to get it. So they have not been able to synthesize this. So commercially made by a fungus, but it's still made in a lab. So we're still going to call it a growth regulator. So site of production in the plant, the shoots, the tips, apical meristem, just like the Auxin. The young expanding leaves, once again, they're just they're produced throughout the plant, same as the other hormones, but they're going to have a regulatory or a balance with the others. The embryo fruits and all of that, which is the same for the Auxin, and some of the tubers or the modified stems for the plants that we'll talk about later on. So what happens with Gibberellins on a plant? It's going to stimulate the elongation of the cells. That's going to be very important. So the Auxin is going to generate the new roots and new cell sites and new callus, and then the Gibberellins will extend it, eventually developing into the roots. There's going to be many plants that are going to be dwarf, and we have them in horticulture. Many of those dwarf plants have a naturally occurring mutation where the receptors are not going to be able to recognize the Gibberellin. And so by not recognizing or able to use the hormone, they're not going to grow properly. Or they're going to have a mutation where the plant may not be producing Gibberellin or the natural hormone, and so it's not able to grow properly or grow to the right height. So many dwarf plants or plants that are sold as dwarfs, if you take an artificial Gibberellin and you spray them, they will become or they will grow to a normal size as any of the other plants. So it's whether a mutation occurs in the plant or it's not able to manufacture this hormone that causes the dwarfism on many of the plants. So it applies to grapes to elongate the peduncle, because as the grapes become mature and as the grapes ripen, they are subject to a lot of the grape molds that tends to kill many of the grapes. So by elongating some of those clusters, they allow for better ventilation and prevents the rotting of the grapes. So that's a different use that it has. Promotes the cell division in the vascular system, hence the cell division that is going to be responsible for the new callus and the new roots in your cuttings. And then it promotes the seed germination. So it promotes it because it begins or awakens the seeds, or the removal is what's going to awaken the seeds for germination. So when the seed germinates, it's going to produce the gibberellins that will then create the enzyme that will begin to break down the starch or the endosperm of the plant, and that's how the seedling will then begin to use that energy to carry out the germination process and then the establishment process. So the enzyme produced from the hormones or after the hormone is made. So this is important for making beer because that's where they will take some of the seedlings, the barley, they will put them in a large vat of water. They're going to germinate the seedlings, and as the seedlings begin to change the starch into sugar for energy, that starch that was made by the seedling as it was germinating, that will then ferment into the alcohol that will cause the beer or the drunkenness on people. So that's exactly what happens here. And influences the fruit or the flower development, which is a combination with the auxin. So here's just a couple of plants. On the left, we have one that has a dwarfism. So the leaves and the stems are very close together, so the internodes are not too far apart. In the middle, we have a plant that is growing properly, and on the right, we have one that is very, very elongated. And so this is going to also occur when plants are going to be leggy. They're not growing in the right amount of sunlight. The plant will reach out for the light, and so it's going to produce a higher amount of this gibberellin to stretch out the stems and try to reach the light or reach out for the light. So that's what's going to happen. In the absence of light, there's going to be more gibberellins and less auxin. So here's just a couple more examples. On the left, we have a plant that is growing naturally, and the right one that is not have gibberellin. And the same thing with the fruit. The one on the right was sprayed with gibberellin, and that's why it's more elongated than the other. Here's the example of grapes, where the one on the right has been sprayed, and that has elongated the cluster, and is not as dense as the one on the left side, which will cause rot. And in the germination of the embryo, so this is what happens. So when the water goes into the seed, that's what's going to trigger the awakening of the embryo. The embryo will then produce the gibberellin here, and the gibberellin is then going to move into the starch, and then the enzyme amylase, that is the enzyme that is going to break the starch that is stored in the grain, and that starch is then going to turn into sugar that will then feed or energize the seedling for the germination process. So it's important for the creation of the different enzymes for the plant to carry out its life. So gibberellin, that's what happens. And so that's gibberellin. Cytokinins is going to be the third one. And cytokinins was first discovered in coconut water. And that's why a lot of people like to grow, or instead of using the artificial rooting powder, they say, oh, just dip the cuttings in coconut water. And that's kind of true because it does have some of the hormones. And that's also why they say that coconut water is good for you, because it has more vitamins and minerals or some other plant compounds. Well, it is this hormone that is found in the coconut water that is what you're drinking. But later on, it was found in the sperm of the fish, and that's when they discovered it, and that's where they got synthesized from. So I don't know what they were searching for, but I guess they found it in there, and now we have it commercially available. So cytokinins, The ones that use in horticulture, different forms. Ceatin from corn. That's the one that was first isolated. The one synthetic used for horticulture, benzodiazepine, BA. You might see that acronym or the BA on the rooting hormone, because they might have both of them or all the hormones just because they balance each other. So the site of production is going to be the embryo for this one, the young leaves and fruit, the apical meristems or the roots and the shoots. And then we have what action happens with cytokinins, promotes the cell division, cytokinesis, as you may have read that word, contributes to the enlargement of the leaves. So by combining with gibberellins and expanding the leaves, it will enlarge them. It stimulates the differentiation in the cells. So remember, gibberellins will elongate the cells, and cytokinins will then change them and promote them to turn into the root. So that will be the differentiation. So remember, we have to take those cells, return them to an embryonic stage, and then allow them to differentiate into the new role. And so that's where the hormone will become important. Combining it with auxin delays the dropping or of the leaves by keeping the chlorophyll production. And so here is the combination of what happens. So when we have a plant on the left drawing that has the apical meristem, the apical meristem is going to be dominating the growth, and it's going to keep the buds on the side or the lateral buds dormant by pumping them with auxin. So the auxin is going to keep the cytokinins at bay. But the moment that somebody cuts the apical meristem, now the auxin is not going to be as high concentration. Now the cytokinins is going to begin to elongate and wake up the lateral buds, and now you're going to have a fuller plant. So the two together, and one inhibits the other, or they balance each other. And so in the same manner as the apical meristem keeps getting taller or higher and moves away from the bottom, then the dominance begins to dwindle because there's less auxin, and then more cytokinins becomes the dominant, and then the lateral buds begin to grow towards the bottom and then at the top, and that creates the Christmas tree shape on many plants. So that's the combination of the two that allows and or prevents or keeps the apical dominance on the plant. And so what we have here is just a drawing of what's going to happen later on in tissue culture. So tissue culture, we have a combination of the two hormones and or several hormones. And this is where a micrometer or micro amount, milligram, can make a big difference. And for a very long time, many labs, when they kept working towards developing the right amount of the different hormones to be successful with growing plants, they will held their a secret, their recipe a secret, and they will not allow anybody, because they can control the market by being able to propagate millions of a specific plant. And so you have going to have an agar, which is going to be a gelatin that is going to have the vitamins and minerals and the hormones that the plant will need, as well as sugar. And so it has everything that the plant will need. And so here's just an example. You take an eggplant, which is going to be a piece of the plant, and the nutrient agar. And then when you look at the amount of the auxin and the cytokinins, when you have no auxin, then there's not going to be any change or no major change. When auxin is then allowed to go in, then that's going to begin the growth of the callus. So this is just going to be a blob of cells that are going to be just embryonic. And then when you increase some of the auxin, then you're going to have the development of the roots, or the roots will begin to grow. And so this is going to be a time process where many plants will have the root first, and then they'll transplant it into a different media that will have now a higher concentration of cytokinins, and then it's going to start to develop the shoot. And once you have the root and the shoot, that is now going to be a full plant. And for all purposes, now we can transplant it and sell it to the public. And so this is where just a different combination and minute amounts is going to make a big difference of what happens or how the plants are going to respond. Ethylene is going to be now the destructive hormone. Ethylene was discovered in the old days when there was different types of oil lamps. There was no electricity, and so every night somebody had to go along the streets and light up every single lamp to illuminate the dark. And so what they started to notice is that any kind of branch from a tree that was growing towards this lamp would all of a sudden begin to defoliate. So they will start dropping the leaves, and it was only those branches that were in close proximity to the lamps. The rest of the tree or the rest of the plant would be good. And so they found out that maybe something within the oil was causing that defoliation to occur. And sure enough, that was the ethylene hormone that was causing that. So ethylene is going to be a gas. Ethylene is also going to be the ripening hormone. So bananas and many green fruits will be gas with this hormone in order for them to induce the ripening and to then eventually be able to sell them out to the public. So it can be a gas or even in a gas state. So that's going to be the form, or it's going to be in a liquid that you can then pour it into a humidifier, and then it becomes airborne, and you can gas or get them all over your plants. The site of production of ethylene is going to be throughout the plant. And ethylene is always going to be produced on rotting plant parts. And so last time we met, I asked for you to go to your flat and remove any leaf that has fallen, remove any cutting that is dead, that was not successful. Keep your flat very, very clean, because having leaves rot on top of your flat or in your flat is going to release some of this ethylene that is going to prevent the rooting or make your plants. Ethylene is also going to rot the flowers, the cut flowers. So if you buy cut flowers, many times they are sprayed with a potassium or some product that will keep the ethylene from reaching them and extends the life of that flower for several days or several weeks. So it's trying to keep them from happening, or then you also change the water and keep them from rotting, because all of that is producing this hormone. So every single plant, leaf, stem, as it rots and dies, it's going to produce the ethylene. And the famous saying that a rotten apple spoils a bunch is exactly right, because the rotten apple is going to release ethylene and is going to hasten or speed up the mature or the ripening of the others until they all going to rot. Or if you have some green avocados, you put them in a paper bag and you add the apple core to speed up the ripening process. So it is the ethylene that is being released by that apple core that will ripen the avocados and or rotten the fruit. So that's going to be throughout the plant. What action is going to have? Inhibits the roots, inhibits the shoots by blocking the auxin from the apical meristem. So again, it's going to have a balancing action on the other hormones. So that's going to be how it's going to work. Stimulate the leaf curling, or what is known as epinasty, that will eventually then drop. So you don't want that to happen. And so the actions of ethylene in the plants, what's going to happen? May induce advantageous roots formation by blocking the other hormones in a cutting, or it enhances the flow of latex in rubber trees. So this is what's some of the other actions that may have. Stimulate the abscission of leaves. So yes, the ethylene will play a role when the leaves are dropping from the tree. And it's maybe useful for dropping some fruits or forcing them to drop and not having somebody go on a ladder and pick them. So for harvesting fruits, that could be useful. Promotes the ripening, apples, tomatoes, and a bunch of others, including bananas. Promotes the senescence, which means the killing of flowers or dropping of flowers. So the fruit stop, the product that is known for, or that is sold for preventing olives from having fruit or liquid ambers from having fruit, is an ethylene-based product. So that's by, it aborts the flowers. And then it enhances the flowers, some pineapple and or bromeliads. So the common bromeliads that you get from the store, especially the baby ones, they are gas and they are forced to flower very early. Or if you buy a pineapple and you grow the top, as long as the plant has enough roots, you can then put a rotten core of apple in the center, and that will force that baby pineapple to grow a new pineapple. So you can have your very own baby pineapple if you want. So in certain plants like bromeliad, it will promote them or encourage them to flower. In a few other plants, it'll force them to drop. So depending on the plant. So here's the action of the leaf drop. So we have the auxin that is going to be free flowing from the apical meristem to the leaves, and as long as it's flowing, the leaf is going to have the full chlorophyll and is going to work properly. As the temperature changes, the increase in the ethylene is going to block the auxin, and then when that happens, then the plant is going to promote the creation of that abscission layer, or the layer that will naturally separate the leaf from the stem. And then with time, the leaf is going to drop. Because auxin is also important for the chlorophyll, once the auxin stops flowing, then the leaves may turn yellow or orange or different color, because now the dominant color is no longer the green. And so some of the other colors that were in the leaf but hidden or masked by the dominant color would then be shown, and that would be the yellowing of the leaf as they drop. So combination of the two hormones. When sprayed on some plants like Gesneriads or African violets, it's going to kill them. So it could have a very negative consequence on certain plants. When sprayed on tomatoes, it's going to kill them. So it's just going to distort them, curl the leaves, not very happy. And I mentioned on bananas. So depending on the company, some of them are going to have an ethylene packet, like a cloth, and they'll put it in wherever they harvest the bananas, so that they are being gassed as they are being brought or transported. So when they arrive here, then they'll send them to the market. Or when they get to downtown LA, there are going to be the wholesale produce area that can also put them in a chamber, and they'll then gas them. The ethylene gas smells like ripe banana, so that's actually the smell. And the pineapple, can they induce it? Not really induce it, but you can induce it to develop or mature. Or sometimes when you buy a fancy box of pears, it's going to have an ethylene absorbent product. So it's the opposite. So they don't want the pears to rot very quickly. And so it's going to, they put a product such as this that will be absorbing the ethylene as is made by the naturally ripening of those pears and apples and a bunch of other things. So those fancy pears will last longer because the ethylene is being removed from the box. So mangoes can also be induced to ripen, and so would cucumbers and or zucchinis or squash. So clean your flat. So that's very important. Abscisic acid, there's not too many things about it. This is just kind of like the forgotten hormone. So similar to gibberellins, they produce producing a plant in all the organs, the roots, stems, flowers, and fruits. What's it doing in a plant? It counteracts the effects of auxin and gibberellin, so it's more like a balancing hormone. Maintains the dormancy in seeds and buds, and so when a seed germinates, when the water is able to penetrate the seed coat, then the water is going to leach out the hormone or this hormone. And so when it leaches out the hormone, then that seedling is then allowed to germinate. So that's why as long as there's no water flowing or inside the seed, then the higher percentage of that hormone is going to keep that seedling dormant. So that's how that's going to occur. Stimulate the guard cells for conserving water. So in photosynthesis, when the guard cells or the stomatas are open, it is the hormone that will open or close them. And so when the plant is going to be suffering or suffering from water loss or water stress, then it is this hormone that will go and close the stoma so that the plant does not lose water and or will desiccate and it will die. And so here's just the stomata. So we have the hormone that is going to be produced inside and is going to be carried to the guard cells, and then it's going to either open them or close them depending on the amount of water that is available in the root system. And just to summarize, so last slide here is just probably the only slide that you should be familiar with. The rest is just a lot of generic information, but just the different part of the plant and just the different hormones and what effect is just having. So control of cell division and Gibberellin on the stem, stomata closing and or closing or opening, abscisic acid on the leaves, control of the stem elongation, the Auxin and the Gibberellins, Auxin and Gibberellic acid. And so I'll let you take a picture. So I think that is the last slide. So once again, should you know all the hormones, you should know the name, be familiar with this last slide, just so that for your information. But I'm not going to ask you to give me an explanation of the hormone or how it works and all of that. Just know that there's those 5 hormones that may have a detrimental effect on propagation, because that's what we're here for, and kind of be familiar with how each one is influencing the cuttings in the production or inhibiting of the roots and all of that. So are there any questions from the audience in the computer? Or questions in the audience?
No, none here.
All right, so then I'll stop the share. So I'll post this one later on. And